Hello and welcome everyone to episode 14, Remastered. In the last episode, I discussed the various genetic experiments conducted by Gregor Johann Mendel, and how his work provided the basis for the study of genetics. It opened it up as a field of science. Now, the principles that Mendel came up with, specifically the principles of segregation and independent assortment, it turns out that these could be explained through meiosis. The scientists Theodore Boveri and Walter Sutton published this hypothesis in 1902, although they were far from the first scientists to study and publish their findings on meiosis. Meiosis as a process was first discovered in the late 1800s by zoologists who were researching eggs. The first discovery came in 1876, when a German zoologist named Oskar Hertwig described meiosis in sea urchin eggs. The second discovery came in 1883, when a Belgian zoologist named Edward van Beneden studied the chromosomes of the eggs of the Ascaris roundworm. It's really not that surprising that meiosis was discovered through the study of eggs, which are one of two types of gametes produced by meiosis. The gametes are haploid, produced after DNA recombination between the homologous chromosome pairs, and then the separation of the homologs into the individual gametes, or a halving of the parent cell's chromosomes to get just one copy of the total genome. This is, of course, the process of meiosis, which I'll discuss in greater detail in this episode. Now, where else should I start but with the chromosomes themselves? The process of meiosis is, after all, the process of the chromosomes of a single cell being equally separated into four gametes. So what are chromosomes? They're basically the superstructure of a DNA molecule when it's wound up tightly into a packaged format. Understand that the DNA molecule is basically one very long string. Each of your cells has 46 string-like molecules of DNA. 23 of these come from your father, and 23 come from your mother. Anyway, each of these 46 DNA molecules has a different length, and when coiled and compacted down into a dense chromosome, they all have different sizes. For that matter, specific chromosomes have bulges and rings, or places where the DNA is packed looser or tighter. Looser packing makes a bulge, tighter packing makes a condensed ring. I should mention that chromosomes are also heavily composed of proteins, which facilitate the proper movement, wrapping, and organization of the DNA as it gets wound up into the chromosome. Most of the chromosomes are autosomes, meaning that they're non-sex chromosomes. In fact, 44 of our 46 chromosomes are autosomes. The remaining chromosomes are the sex chromosomes. And so there's only one sex chromosome per set of 23 chromosomes in the genome. And because you have two copies of the genome, you got 46 chromosomes, you have two sex chromosomes. Female humans possess two copies of an X chromosome, while male humans have one X chromosome and one Y chromosome. These sex chromosomes contain genes that code for our respective genitals, the physical development of the ovaries, uterus, and vagina in females, or the physical development of the testes, prostate, and penis in males. All of these structures require specific gene sequences, which code for various proteins and regulate their expression, and thus their activity, and the biological structures that they produce. These genes are located on the sex chromosomes, although it should be understood that other genes exist on the sex chromosomes that have nothing at all to do with the genitals or the reproductive systems. Now, all of these genes on the sex chromosomes, whether or not they have anything to do with the genitals, all of these genes are called sex-linked genes. They're linked to the biological sex of the individual in that their specific sex chromosomes influence the gene's expression. A gene that's in a Y chromosome, for example, will only be expressed in males, as females don't have a Y chromosome by definition. They don't have one in the first place. Okay, so we have 46 chromosomes, 23 coming from each of our parents. A chromosome in one parent corresponds to a chromosome in the other parent. These similar or homologous chromosomes contain genes that code for the same traits, but they might have different alleles. A useful name for these chromosomes is homologs, or homologous chromosomes, where the prefix homo implies the similarity of the genes. Because humans possess 2 copies of our genome, we are a diploid species. Many other species are also diploid, possessing 2 copies of their genome in their cells. But some species are haploid, possessing only 1 copy of their genome. Gametes are cells that are produced by sexually reproducing diploids, but the gamete cells themselves are haploid. Now, other species may even have triploid or tetraploid cells, having 3 or 4 copies of their genome. Although this particular genetic arrangement seems to be much more common in plants than in anything else. The thing that creates differences between our mother and father, and differences between every individual human, is our specific combination of alleles. The allele is a version of a gene. An example that I like to use is eye color. All humans have an eye color. We all have genes for the eye color trait. The allele is just the version of that trait. So for the eye color gene, one allele might code for green eyes, while another allele codes for brown eyes, and a third allele codes for blue eyes, and so on and so forth. A large minority of the genes in the human genome have numerous alleles, just like the genes that code for eye color or the genes that code for the level of pigment in your skin. These genes usually code for traits that are cosmetic or open to variation. I know this isn't a cosmetic example, but consider your blood type. If you're type O, then you have alleles that code for the plasma antibodies anti-A and anti-B, but none for blood cell antigens. If you're blood type AB, then you have alleles that code for both the A and B antigens that coat your red blood cells, but you have no alleles that code for plasma-dwelling antibodies. If you have blood type A, you have alleles for the anti-B antibody in your plasma and the A antigen coating your blood cells. And lastly, blood type B is the opposite, possessing alleles for the plasma antibody anti-A and blood cell coating B antigens. For the rest of our genes, for most of our genes, we all have the same alleles. These genes tend to code for really important things, like respiratory proteins, or proteins involved in fetal development or brain function. If we didn't have these genes, or if there was a mutation to these genes that disabled the protein or altered the gene's expression, we would almost certainly die. Mutations to these highly conserved genes result in offspring who, for example, aren't able to breathe properly due to a mutation in a respiratory protein, or who develops improperly in the womb and becomes stillborn, or who has some neurodegenerative disease because of some protein failure in his brain. All of these highly conserved genes basically code for proteins that are extremely important to basic homeostasis or some fundamental life process. Other examples of such highly conserved genes include those that code for proteins involved in the electron transport chain, or neural tube development, or the mouth-nose complex. Because of the high risk of mortality associated with any kind of mutation here, the functional versions of these genes are highly conserved through natural selection. Many of these highly conserved genes have just 1 or maybe 2 alleles, or a small handful of alleles that have extremely small differences between them. All genes, whether they be highly conserved genes or highly mutable genes with many different alleles, all genes are heritable. This is to say that all genes can be passed down from parent to offspring. The traits that the offspring receives are those that are coded for in the specific alleles and the epigenetic patterning carried by the fertilizing gametes. So when a sperm binds with and fertilizes an egg, they mix their haploid genetic material together to create the complete diploid genome of a new person, of a new individual. This genetic material takes the form of chromosomes. Now, before I cover the process in depth, I want to briefly summarize the 2 stages of meiosis so you get an overview of where we're going as we're charting the seas. The first stage is very similar to mitosis. The DNA condenses, forming 46 chromatids. Each chromatid is a single string of DNA that's been compacted and coiled into the chromosome superstructure. The 46 chromosomes will then associate with their homologue, creating 23 pairs of homologous chromatids. Each chromatid will then replicate itself. Each homologous pair is now composed of 2 groups of 2 DNA molecules, for a total of 4 chromatids. This forms a complex called a bivalent. The 23 bivalents line up at the metaphase plate, just like they do in mitosis, where they get split up and separated into 2 corners of the cell. Once the cell divides and produces 2 diploid daughter cells, this stage of meiosis is over. And understand that these daughter cells are diploid because the parent cell at the beginning of Meiosis One geared up with a total of 4 copies of each chromosome. So when it divides the first time, each daughter cell gets 2 copies. If this were mitosis, these daughter cells would then mature and go on to live functional lives. In meiosis, however, these daughter cells undergo another round of division called Meiosis 2. So understand that the process as I describe it, the process of Meiosis 2, is happening to both of the daughter cells that were produced from Meiosis 1, and it's happening to them at the same time. Meiosis 2 is similar to Meiosis 1, in that the DNA is once again lined up in the middle along the metaphase plate, where it's split up and dragged to opposite ends of the cell, and the cell then divides. The critical difference here is that in Meiosis 2, the DNA doesn't get replicated beforehand. So in Meiosis 1, we have 23 bivalents, or 23 groups of 4 chromosome copies, each of them being split apart into 2, and into pairs. This resulted in 2 daughter cells, each with 23 chromosome pairs, or 46 chromosomes in total, so they were diploid. Now in Meiosis 2, this initial DNA replication doesn't happen. There's no bivalence in Meiosis 2. Instead, the structures that get pulled apart are composed of just 2 chromatids, and when they get split up, only 1 chromosome gets pulled to a corner of the cell instead of 2. As a result, the daughter cells of Meiosis 2 only have 1 copy of the chromosome. In this way, mitosis takes 1 diploid parent cell and produces 2 diploid daughter cells. In meiosis, a diploid parent cell produces 4 haploid cells. These haploid cells are called gametes, and they are the fundamental key to sexual reproduction. With that brief summary out of the way, let me start back at the beginning, at the start of Meiosis One, so that we can explore the mechanisms and details of the whole process a little bit slower and with a little more granularity. Meiosis One is itself composed of 5 phases, which are called in sequential order Prophase 1, Metaphase 1, Anaphase 1, Telophase 1, and lastly, cytokinesis. Prophase 1, the first and longest stage, can also be broken up into substages, namely the leptotene, zygotene, pachytene, diplotene, and diakinesis stages. In early Prophase 1, specifically in the leptotene stage, the cell is fresh out of interphase, fresh out of the G2 stage of the cell cycle. To mark its entrance into M phase, the DNA in the cell begins to get packed and condensed into chromosomes, and the spindle apparatus and its associated complex of microtubules begin to form. The synaptonemal protein complex that holds homologous chromosomes together also begins to form. A fundamental and defining aspect of meiosis occurs early in Prophase 1, during the zygotene stage, where the homologous chromosomes associate with one another, naturally forming those 23 pairs of homologs. This natural association between homologs is called synapsis, and it doesn't occur in mitosis. The cell undergoes synapsis when its homologous chromosomes physically associate, lining up side by side with one another. Now, another important event happens immediately before this pairing. A deliberate break is induced at some random point in the DNA strand of a chromatid. This break in the DNA will be involved in a future crossing over event, but I'll get to it as it happens during the whole process. Anyways, this synapsis forms bivalents, which are, as I said, two sister chromatids associated with their homologs, which are themselves sister chromatids. Because there's 4 strands of DNA involved in a bivalent, they're also called tetrads. The bivalent and all of its chromatid copies are held together by a synaptonemal complex, which is a group of remarkably adhesive proteins. The formation of the bivalents, or the tetrads, brings us into the Pachytene stage. This event is marked by the collapse of the nuclear envelope. Both phospholipid bilayers in the double membrane of the nuclear envelope begin to dissolve, falling apart in clumps before ultimately melting away into the cytosol. The microtubules that were formed earlier in prophase 1 begin to randomly attach to the kinetochores of the chromosomes. As the microtubules attach, the synaptonemal complex begins to collapse, and the bivalent loses its structural integrity. This causes the homologs to pull apart, but they don't separate entirely. When some parts of the homologs detach, they form gaps called chiasma, or chiasmata, while other parts of the homologs remain stuck together. These points where the DNA sticks together are the same points that experienced DNA strand breakage earlier, at the beginning of prophase 1. Most homologs get at least 1 chiasma during prophase 1, although 2 or 3 chiasma on a single homolog pair isn't unheard of. So it's at these points of residual sticking together that exchanges in the genetic information begin to happen. The maternal and paternal homologs physically swap portions of their DNA with one another in a process called crossing over. Crossing over is a really important event because it generates genetic diversity. You receive discrete chromosomes from your mother and father, but what happened was your mother's and father's chromosomes, their discrete structures, exchanged parts. They switched it up a little bit. And so the discrete chromosomes that were produced each had little bits of the mother and father chromosomes. And so each individual chromosome that was newly created in this recombination is unique. There's a genetic uniqueness to it, and so this creates genetic variety. Crossing over, or recombination, causes your homologs to shuffle up a little bit, so that a unique allele combination is passed down into your offspring. It'll be why your kids look like you, but not exactly like you. And it's also why your kids will look different from each other, even though they're all being born to the same 2 parents. Anyways, these chiasma form and enable crossing over to occur in the Pachytene stage. And this, in addition to the collapsing synaptonemal complex, allows the homologous chromosomes to begin to separate, which defines the diplotene stage. In the diakinesis stage, the chromosomes have condensed enough to make themselves and their chiasmata visible under a microscope. Additionally, the meiotic spindle begins to form as the nuclear envelope collapses. Pairs of centromeres push into opposite corners of the cell, with their microtubules reaching out for the chromosomal kinetochores. Once the microtubules from the spindle apparatus have attached to the chromosomes, the cell will enter Metaphase 1. In this stage, the spindle apparatus organizes all of the tetrads into a single plane running along the center of the cell, called the metaphase plate. In this way, the metaphase stage is very similar in meiosis and mitosis. The cell will now enter anaphase 1. The microtubules begin to fray, pulling chromosomes to the centromeres of the spindle apparatus situated at each end of the cell. The new homologs in each tetrad are pulled apart by the retracting microtubules, and the cell enters telophase 1. Here, the retracting microtubules drag the separated homologs to each end of the cell. Now keep in mind that the sister chromatids, even though they might have been altered a little bit by recombination, they're still attached together. The last stage occurs when everything has separated. Cytokinesis occurs as the cytoplasm is pinched off to form two new daughter cells, each with freshly mixed chromosomes. The second stage of meiosis is unimaginatively called Meiosis 2. Each daughter cell of Meiosis One goes through Meiosis 2 to produce two daughter cells of their own. So after the process of meiosis, one progenitor cell will produce 4 daughter cells, or 4 gametes. Now Meiosis 2 also has 5 stages: Prophase 2, Metaphase 2, Anaphase 2, Telophase 2, and again, cytokinesis. These stages are all very similar to their analogous stages in Meiosis 1. Prophase 2 will begin with the quick reformation of the spindle apparatus. If the nuclear membrane was partially or fully restored after Meiosis 1, it dissolves again during Prophase 2. Recall that the daughter cells of Meiosis 1 each have 2 copies of the genome, but most or all of these chromosomes have new combinations of alleles from the maternal and paternal chromosomes crossing over. This means that Meiosis 2 involves cell replication with half the number of chromatids of the previous replication. The 23 pairs of recombined sister chromatids will attach to the newly formed spindle apparatus. And this takes us to Metaphase 2, where the spindle apparatus organizes the sister chromatids along the metaphase plate. During Anaphase 2, the centromeres of the spindle apparatus are pushing into opposite sides of the cell, and they're pulling all of the chromatids with it. This combined force causes the sister chromatids to be physically pulled apart. And this also causes the cell to enter telophase 2, which is a brief stage where the microtubules once again fray at their tips, dragging the attached chromatids back towards the centromeres. Once each set of 23 chromosomes has been pulled to opposite sides of the cell, the membrane pinches off and forms 2 daughter cells. As Meiosis 1 produced 2 daughter cells, their combined replication in Meiosis 2 produces a total of 4 daughter cells. But notice that these final 4 daughter cells only got 1 set of 23 chromosomes, not 2. This makes these final daughter cells haploid, and this is the defining characteristic of the gamete, or the egg and the sperm. In this way, the gamete-producing cells in your ovaries or your testicles begin as a single diploid cell with replicated chromosomes, which divides into 2 diploid daughter cells, who each then divide into 2 haploid daughter cells. These haploid daughter cells, or gametes, are egg cells in females and sperm cells in males. As haploid cells, they only have 1 copy of their chromosomes, so when a sperm fertilizes an egg, the cells will chemically merge together. Their precious DNA will meet inside of the egg. It'll bind together and it'll initiate the chemical processes that translate and transcribe this now full set of DNA to produce a new human being. It's important to understand that this really is a new human being. Asexual organisms have plenty of offspring, but they're all genetically identical. Sexual reproduction, or the use of gametes to shuffle allelic information, is able to generate a huge degree of genetic variety. This genetic variety is healthy for the gene pool, as it induces greater phenotypic variations in the population. In the wilderness, maybe not so much in our sheltered and comfortable urban lifestyles, but in the wilderness, greater phenotypic variation meant that a species had greater evolutionary adaptability. If there's a wide variety between individuals, it's more likely that some of them will survive exposure to an adverse condition, like a disease or a drought. A great deal of this variety is generated through independent assortment, where each chromosome is pulled into one daughter cell or the other, independent of where the other chromosomes are going. To put it another way, groups of associated genes on different chromosomes are not passed down as a consistent collection of alleles. Independent assortment shuffles the chromosomes randomly, so that the combination of alleles in an offspring is different than the specific combinations of alleles in either parent. If you want to try and get a glimpse of how much variation this can produce, take a peek at the math behind it. A diploid species has 2 copies of their chromosomes, so they have 2 to the power of n possible combinations of maternal and paternal chromosome sets, where n is equal to the number of chromosomes in a single set. As humans have 2 copies with 23 chromosomes in each set, the number of possible combinations of maternal and paternal chromosomes is 2 to the power of 23, or 8,388,608. This is just the variety that's possible in a single offspring of 2 parents. When you start taking into consideration the possible genetic combinations of families or entire lineages, the number of possible combinations becomes astronomical. The actual flesh and blood people depicted in dusty history books and extended family trees are the single combination out of that 8.4 million that happen to make it, that happen to get fertilized and become manifest in the biological substrate of flesh and blood. For that matter, you, as you are right now, are the one single combination of genes and alleles out of the 8.4 million possible combinations your parents could have had. They could have had any kid out of the 8.4 million possibilities. And for better or for worse, they got you. Now that I've talked about the process of meiosis as it properly occurs, I want to talk about what happens when stuff goes wrong. As meiosis involves the DNA itself of an emergent life form, the proper unfolding of the process is absolutely critical to its health and its vitality. Even the smallest mistake this early in the development of a new organism will almost always cause catastrophic and usually lethal malformations or damages. At a minimum, 25% of human conceptions are spontaneously aborted when errors or problems in meiosis are detected. So every time a woman becomes pregnant, there's a 25% chance that that embryo is just going to spontaneously self-abort. This is technically a miscarriage, but when it happens this early in the pregnancy, the woman may not even notice. She may not even know that she was pregnant in the first place. You might have gotten the impression that meiosis is a delicate process, and it really is. It has several steps that have to be conducted pretty much flawlessly, especially steps involving separation of the chromatids. Failure for the chromatids to properly separate is called Nondisjunction, and it can be a very serious problem. The homologs have to separate properly during Anaphase 1, such that each daughter cell gets only one homolog from each of the 23 pairs. If they don't, one daughter cell will have too many homologs, and the other daughter cell will have too few, and this will complicate subsequent steps and produce gametes with an irregular number of chromosomes. Another important event is the separation of sister chromatids into each gamete in Meiosis 2. If the sister chromatids fail to separate, then both of them will get dragged into a single daughter cell. Gametes with irregular numbers of chromosomes produce offspring with irregular numbers of chromosomes, and these offspring suffer from the associated genetic diseases and abnormalities. So let's say that some chromatids suffer nondisjunction. That is, they fail to break apart properly. The emergent gametes have an irregular number of chromosomes. One has too many, and the other has too few. If the gamete with too few chromosomes can become fertilized with a healthy normal gamete, then the emergent zygote will be missing one copy of a chromosome. This condition of having a single copy of a chromosome is called monosomy. Conversely, the gamete that has too many chromosomes can become fertilized with a healthy and normal gamete, and this will produce a zygote with an extra copy of a chromosome, and this is a condition called trisomy. The tri in trisomy designates that the zygote has 3 copies of that chromosome, instead of the normal 2. When an organism has any of these irregular chromosome counts, it's called aneuploid. For a well-known example, consider chromosome 21. If a human zygote is aneuploid with only a single copy of chromosome 21, so it suffers from monosomy 21, it virtually never survives until birth. On the other hand, if a human zygote is aneuploid with 3 copies of chromosome 21, it will suffer from trisomy 21. Zygotes with trisomy 21 are much more likely to survive till birth than those with monosomy 21, but they suffer from Down syndrome. Down syndrome is characterized by delayed growth and cognitive development, intellectual disability, and several distinguishing physical characteristics, including short height and finger length, slanted, wide-set eyes, and a large tongue that can induce obstructive sleep apnea. The condition of trisomy 13 is known as Patau syndrome, and it's a brutally disfiguring genetic disease. Those with trisomy 13 who happen to be born alive will suffer from microcephaly, or a shrunken head. They'll also suffer from a cleft palate, from eye and spine defects, and brain disabilities, as well as abnormal numbers of fingers and toes, deformed feet, hands and genitals, heart defects, and kidney problems, among many other symptoms. Unfortunately, this severely disfiguring disease is extremely difficult and expensive to treat. 80% of children with trisomy 13 die before they turn 1 year old. Consider chromosome 18. Trisomy 18 is known as Edwards syndrome, and it's a similarly disfiguring genetic disease. Symptoms of Edwards syndrome include heart, lung, brain, and intestinal malformations that lead to impaired growth and difficulty eating and breathing, as well as brain cysts that form in a layer called the choroid plexus. The head is usually small, with low-set ears and a small jaw under a cleft palate. The hands are typically underdeveloped, missing fingernails. The feet are malformed, and this impairs proper movement. Now consider trisomy X, also called triple X syndrome. Now, unlike the other trisomy diseases that I've just mentioned, trisomy X isn't brutally disfiguring or highly lethal, and many women who have it are still fertile and able to have healthy children. Affecting about 0.1% of women, possessing 3 X chromosomes can induce the formation of subtle but distinguishing characteristics, like wide-set eyes and above-average height. Trisomy X also isn't nearly as psychologically and intellectually disabling as the other examples. Some women with trisomy X experience slow language development, seizures, depression, and anxiety, but these symptoms aren't super widespread and they're not universal to the entire population of women with trisomy X. In a much more general sense, trisomy is a condition that affects smaller chromosomes, like chromosomes 13 to 22, much more often than it affects larger chromosomes, like chromosomes 1 to 12. With this in mind, it makes sense that trisomy 21 is the most common kind of survivable aneuploidy, as chromosome 21 is the smallest of the human autosomes. Furthermore, these problems are far more common in children born to mothers over the age of 35, due to the fact that the biomolecular machinery in the eggs begins to degrade in both structure and function over time. That about covers it, I believe, for meiosis. It's important to understand that meiosis came about in evolutionary history as a mechanism for generating genetic diversity. Genetic diversity confers variation in the phenotypes, and thus variation in fitness of individuals within a population. This phenotypic variation makes the population better able to survive and adapt to strenuous conditions and selection pressures. Species that reproduce asexually do not have this built-in means of creating genetic diversity. An asexual individual will pass all of their genes on to all of their offspring, including any deleterious or mutated genes. Asexual populations are thus at a greater risk from diseases, as they often lack the genetic variety to defend themselves and to survive a disease outbreak. As a result, a single disease can easily wipe out an entire population, and that can have downstream repercussions. Sexually reproducing species can be wiped out by diseases too, no doubt about it, but from the perspective of the disease-causing pathogen, it's a lot harder to take out a sexually reproducing species as it is to take out an asexual species. In an asexual population, every individual has the same chemical defenses, so you overcome the defenses once, and you overcome them for every castle in the kingdom, so to speak. But if you're attacking a sexual population, every individual has a slight variation in their immunology that gives them variable defenses to the disease. So, to continue the metaphor, it's like every castle in the kingdom has a slightly different defense, and as the attacking pathogen, as the invading force, you have to work your way through each slightly different defense to get into each castle. It's a little harder, and it'll take you a lot longer. When you're looking at the evolution of life and the variables that will increase your likelihood of survival, this increased genetic variety is extremely valuable. Extremely valuable. It cannot be understated how valuable it is. I mean, to put this in perspective, consider that it took over 1 billion years to evolve multicellularity. It took over 1 billion years for life to move from single cells to organisms that were composed of multiple cells. And then consider the fact that in less than half of that amount of time, these multicellular species that evolved sexual reproduction and the genesis of genetic variety with every generation, in less than 600 million years, this has generated all of the biodiversity you see around you. All of the trees and flowers and grasses and all of the other land plants, all of the funguses, all of the birds and the bees and all of the other animals, the primates, mammals, fish, amphibians, reptiles, all of this came about in the last 500, 600 million years. And the raw speed with which all of this speciation and divergence has gone to work can be fundamentally attributed to the genetic variety produced through meiosis. Well, I think that's about it. I hope you thought this episode was cool, and I hope you learned something that really got your brain working. Something that just really tickled your mind. And as always, thanks for listening.